അതുൽ ജോയിൻ ചെയ്യാൻ പറ്റുമോ?
ഇല്ല മാം, അതുലില്ല.
Recording in
ഓക്കെ അതുൽ, ഞാൻ ജോയിൻ ആയിട്ടുണ്ട്. ഓക്കെ ഓക്കെ. Hello everyone. So we are starting, already late by 10 minutes. Okay. അപ്പൊ ഇപ്പൊ തന്നെ അവർക്ക് എല്ലാവർക്കും യൂട്യൂബ് ലൈവ് കാണാൻ പറ്റുമോ സ്ട്രീമിംഗ് ചെയ്യുമ്പോൾ?
യെസ് മാം, അവർക്ക് കാണാൻ പറ്റും.
ഓക്കെ, അതിന് ഡയറക്റ്റ് കാണുമല്ലേ?
അതെ മാം, അത് ഡയറക്റ്റ് നമ്മൾ സാധാരണ യൂട്യൂബ് ലൈവ് കാണുന്നത് പോലെ കാണാം.
ഓക്കെ ഓക്കെ. എന്തെങ്കിലും ഇഷ്യൂസ് ഉണ്ടെങ്കിൽ പറയണം കേട്ടോ. അപ്പം ഞാൻ ഇടയ്ക്ക് എന്തെങ്കിലും അസൈൻമെന്റ് ആയിട്ട് തരാം, അത് നിങ്ങൾ ഹിസ്റ്റോ ഗ്രൂപ്പിൽ സെൻഡ് ചെയ്താൽ മതി. ഞാൻ മാഡത്തോട് പറയാം ഇതിന്റെ ഇഷ്യൂ കേട്ടോ. ഓക്കെ, സോ വി ആർ സ്റ്റാർട്ടിംഗ് വിത്ത് ദ കോർണിയ ആൻഡ് റെറ്റിന ഹിസ്റ്റോളജി. We are only having a limited time. So most important topic in this session is histology of cornea and retina. So the other anatomical aspects you will be learning while you learn the gross part. Okay. But according to a new syllabus, eyelids, lacrimal apparatus, they are also, you should know them. The histology, they will be asked in the written exam. Okay, they can be asked. So that part I'll be just mentioning. Other part you are all having some seminar in the coming week, right? About eyelid, lacrimal apparatus and all. Have you been informed about that?
Yes ma'am.
Yes ma'am. While taking that, you should cover that portion also. Okay, I'll be just mentioning the part. I'll be giving more stress to cornea and retina. Okay, so let's start. So you all know about eye. Where is it located in the bony cavity? You already have seen the orbits, isn't it? Orbit is the bony cavity where it is located inside the which bone? Skull, isn't it? Not a single piece of bone. The different margins are formed by different bones of the skull. Okay, so when we are talking about eyes, it is a peripheral organ for vision. Okay, So you all know the basic structure. Already you have learned in your school classes. It has got a, how many layers for eyes, you know? For the eyeball, how many layers are there?
3.
What are they? What are they? Can you name that?
Clear.
Sclera. Yes.
Choroid.
Sclera, choroid, retina. Okay, you are seeing here sclera, choroid, retina, isn't it? So there are some modification in anterior and posterior aspects that you will be learning in detail. So you know a structure called as cornea, isn't it? That is a transparent part through which the light enters. Okay, that cornea is actually an anterior part of this sclera, which is modified. This sclera, this whitish area, is not transparent. But when it comes in this area, it becomes transparent, and that part is what we call as the cornea. So we have to learn the differences in these parts. Okay, so the next layer, it is the choroid. Can you see blood vessels? So this is nothing but the vascular layer of the eyeball, and you have the inner layer, the retina. That is the neuronal layer. The sensation actually is the photoreceptors are present in this layer, retina. Because of this layer only, we are able to see retina. Okay, then you have some other structures here. What is this? This is lens. Okay, then you have a cavity in front of the lens, the cavity behind the lens. Okay, which fluid is filling all these areas? Here it is vitreous humor. What about this area? What is it?
Aqueous.
Yeah, aqueous humor. Okay, this is the aqueous chamber and this is the vitreous chamber. So for the aqueous chamber, there is two parts, anterior chamber and posterior chamber. That is, anterior chamber is between this cornea and this iris part. Okay, this is the anterior chamber. That is filled by aqueous humor. Then the posterior chamber is not this part. It is actually the part of the aqueous chamber which is behind this iris, okay, in this portion. Can you see posterior chamber marked here? Okay, so this portion, the portion between the iris and the lens. Here there is a chamber. That is the posterior chamber. Both anterior as well as the posterior chamber is filled by aqueous humor. Don't get confused. Don't call this as posterior chamber. This is nothing but the vitreous chamber. Okay, or a vitreous body. Okay, that is about the anterior and posterior chamber. Both of them are filled with aqueous humor. Okay, so we have mentioned about the outer coat. That is the, what are the outer coat? Name the outer coat, quick. Already late. It is sclera and in front by
Cornea.
cornea. Okay, that is the outer coat. What is it a type of? It is a type of fibrous coat. Okay, so the next one, second middle layer, is the choroid. So here the choroid layer posteriorly. So anteriorly it has got some modification here. Can you see this? Okay, this portion. Can you see the laser point?
Yes, ma'am.
Okay, so this is a ciliary body, and from there hangs something. Okay, what is this? It gives a color to your eye pupil. This part. This part. Okay, that is nothing but the iris, which acts as a diaphragm to hold the lens. So this portion, the iris, the choroid body, and the choroid. These are actually modification of the second layer. That is the vascular layer, middle layer, and the inner nervous cord. That is the retina. So what is this structure coming out of this eyeball? What is this?
Optic.
Yeah, that is the optic nerve. That is a cranial nerve. That is in connection with the sensation. Okay, sensory nerve. This is a sensory nerve associated with the vision. Okay, so it carries the impulses from the eye to the brain. Okay, so this is the retina. So we have to study in detail about the histology of this part, the cornea as well as this retina. So just make sure this point. When I mentioned the term posterior pole of the eyeball, look at this. This is an oval shape, right? So this part directly behind this portion is actually the posterior pole. I am not mentioning this portion. Okay, this portion is where you call the macula lutea, yellow spot, all you have studied earlier. Okay, this spot, where the optic nerve emerges, is known as the blind spot. Okay, here there is no photoreceptor cells, but here in this posterior pole or the macula lutea, the center portion of which is known as the fovea centralis, that portion is having the maximum concentration of the photoreceptor cells. Okay, so make sure when I mention the term posterior pole, you should be thinking about this point. And the optic nerve is actually not directly posterior. It is not from the direct posterior pole. It is actually somewhat medial to this fovea centralis when you consider this eye. So if you consider this as your right eye, from there, directly backward will be your, from this pupil directly back, straight behind will be your fovea centralis. But the optic nerve will be emerging small distance medial to this. I mean from the globe. Okay, and will be going medially. Okay, this is about the eye as in a general. So when we are speaking about the structure of the eyeball, I already mentioned the layers. So I already mentioned about the chambers, anterior chamber. You can see it clearly here, the posterior chamber, then filled by, anterior and posterior chamber is filled by what? What is filling anterior and posterior chamber?
Aqueous humor.
Yeah, aqueous humor. So here only comes the vitreous humor, this portion, and this is not the posterior chamber. Okay, this is the posterior chamber behind the iris. Okay, between the iris and the lens, this is the posterior chamber. Okay, so now let's discuss about the outer fibrous core, that is the sclera. So sclera is the outer wall of the eyeball, which is in the posterior 5/6 only. So what is in the anterior 1/6? What is in the
Cornea.
anterior 1/6? Yeah, it is a cornea. So I am just now speaking about the posterior 5/6 only. That is the sclera. This is a thick white opaque membrane. And it is having, I already mentioned, it is a fibrous coat. So what are the fibers? Collagen, elastic fibers along with fibroblast. So it is covered by ocular conjunctiva and episclera. That is, the outer surface is covered by the conjunctiva. Conjunctiva is there two types, ocular and palpebral. Ocular is the part of the conjunctiva that covers the eyeball, whereas the part of the conjunctiva that covers the eyelid is known as the palpebral conjunctiva. So here it will be the ocular conjunctiva only because it is the eyeball. And the inner aspect of the sclera has got an inner aspect also, right? That will be covered by episclera. Okay, posterior by the, that is a facial sheath, that is by the episclera. So the deep surface of the sclera, what is next layer deeper to sclera? It is the choroid. So there is a space in between the sclera and the choroid, that is known as the perichoroidal space. Okay, there is some connective tissue in this space that is known as suprachoroid lamina or the lamina fusa. So the perichoroidal space is filled with suprachoroidal lamina or lamina fusa. That is nothing but connective tissue. So anteriorly, we mentioned it is only 5/6, posterior 5/6. The anterior 1/6 is actually the cornea. You can see here. So this anteriorly, sclera becomes continuous with the cornea at this junction, that is known as the corneoscleral junction, otherwise known as the limbus. So let me show you this picture. Here, this portion where the sclera is continuous with the cornea is known as the corneoscleral junction or limbus. So here where will be that? This junction of this colored area, this round area, junction between this white and the colored area. That round portion is known as limbus. That is very important for you. So make note. This circular margin of this colored portion is known as the limbus. Okay, that is about the limbus, otherwise known as corneoscleral junction. Okay, then a circular channel, known as the canal of Schlemm, you can see here, okay, is present at the junction, at the corneoscleral or the sclerocorneal junction. It is also known as the sinus venosus sclerae, okay, or canal of Schlemm. It is located at the junction. And there, a triangular mass of scleral tissue projects towards the cornea, just medial to the sinus. You can see here this triangular mass of scleral tissue that is known as the scleral spur. Okay, this portion is known as the scleral spur. So I already mentioned about the optic nerve. The optic nerve is attached with a short distance medial to the posterior pole. When you look at the diagram of eyeball, it can be identified that this optic nerve is directed somewhat medially to the posterior pole. Okay. So here, what happens to the sclera there in the posterior pole? Already mentioned the optic nerve have to emerge from the where? From the blind spot or that area, isn't it? So that area, what happens to sclera? The sclera is actually sieve-like, perforated. Okay, that portion is therefore known as the lamina cribrosa. Okay, so optic nerve is attached to the posterior pole of the eyeball, where the sclera is perforated like a sieve and is known as the lamina cribrosa. So what are the functions of sclera? Already mentioned it is a fibrous cord. About the fibrous cord, what will be the function? First of all, protection, maintaining the shape. So inside the eye, we have mentioned many fluids, aqueous humor, vitreous humor. So that will having a pressure, intraocular pressure. It is like a fluid filled a balloon. Okay. So that maintains the shape by the intraocular pressure. And also if it is increasing, if it does not have that much fibrous tissue, what will happen? It can bulge from one side because inside it is fluid. But it is not happening. It is maintaining the shape because of this sclera layer. Then what happens to the external surface? The external surface is smooth. Because of that only, what happens? There is no friction. Our eye is constantly moving. So because of the smooth external surface that is provided by the sclera, there is ease of eye movement. And also, the eye has several muscles attached to that you will be learning in gross anatomy. So that muscles are attached to this outermost layer of sclera. So coming about the cornea. Cornea is the anterior one. This you have to hear more, means understand more clearly because this is a important histology for you. So this anterior one sixth. Is it transparent or opaque? Transparent. Only when light will pass through the cornea, right? So it is transparent. It is convex forwards, isn't it? It is colorless. Okay. Then what gives the color to the eye? Is it the cornea or something else?
Iris.
Yeah, the color of the eye, blue eye, brown eye, black eyes, all are because of iris only. Iris is a thing that imparts color to the eye. Okay, it is avascular. Vascular means what? Does not have blood vessels in that. That is why they are maintaining their transparency. If there were blood vessels passing through the cornea, will cornea be that much clear surface through which, like a glass, can light pass through that if there was blood vessel? No. So for maintaining this transparency, it is vascular. So, but it is having a very rich nerve supply. If you get some foreign body in your corneal part, it is very irritating. So that is very rich nerve supply. Okay. So another important part, what are the layers of cornea? It has got 5 layers. So we have a code for that. Can you note the code for that? I have just highlighted it. What is that? A, B, C, D, E, isn't it? Okay. What is A? That is the anterior-most layer. Anterior corneal epithelium. B, Bowman's membrane. C, corneal stroma. D, Descemet's membrane. And E, endothelium. Okay, A, B, C, D, E. This is the 5 layers of the cornea. You should never forget this. This will be asked anywhere in your, not only in anatomy, it can be asked anywhere. Okay, anterior corneal epithelium, Bowman's membrane, corneal stroma, Descemet's membrane, and endothelium. So, these are the microscopic features of cornea. The first layer, the anterior corneal epithelium. So, that is epithelium. So, what type of epithelium it will be? It is non-keratinized, stratified, squamous epithelium. Therefore, corneal epithelium is a non-keratinized, stratified, squamous epithelium. And the cells in the deepest layer of the epithelium are columnar. Middle layer is polygonal. So you all know how to draw the non-keratinized stratified squamous epithelium. Basal cells will be columnar. Middle layer will be polygonal. Superficial layers will be flattened. This is how you should draw. Okay. Then what about the electron microscopic view? So under electron microscopy, there is some projections in the form of microvilli or plasma membranes or the superficial surface, which helps in retaining a film of fluid. What is the fluid that covers the cornea? What will be the fluid covering the cornea? Forming a film.
Tears.
Yeah, tear film. Very good. That is a tear film. To hold the tear film, there is microvilli, but we can't see that microvilli and all in the light microscopy. It is only visible under electron microscopy. Okay, what about the regenerative capacity? Corneal epithelium regenerates rapidly after damage. So make sure I'm not mentioning cornea, I'm mentioning corneal epithelium. So please make note of that. It is the epithelium, that is the first layer, anterior or the outermost layer, that regenerates rapidly after damage. Why I mention like that? Because there are some layer that comes deeper. If gets damaged, is leaving a scar or will not regenerate. So make sure that is the anterior corneal epithelium is having a very good regenerative capacity. So what is the next second layer? That is Bowman's membrane. Okay, this Bowman's membrane is resting on the corneal epithelium. It is also known as anterior limiting lamina. Okay, what is the other name for Bowman's membrane? Anterior limiting lamina. So there might be some posterior limiting lamina, isn't it? So let's look for that. So this anterior limiting lamina, also known as Bowman's membrane, is structureless when you view it under light microscope. But under electron microscope, we can see dense, homogeneous, fine collagen fibers in matrix. Okay. So what is the function of this anterior limiting membrane? It provides great stability and strength to the cornea, and it acts as a barrier to the spread of infection. Okay. So if there is a minute corneal epithelial aberrations and all, there are chances of spread of infection from a wound, right? Corneal epithelial aberrations are types of wound only. Okay, there also, as we get some aberration of the skin only, cornea is getting aberration. Okay, why we can't see any bleeding if we get corneal aberration? We will be getting some bleeding when we get the aberrations over the scleral part because it is avascular. We can't see any blood oozing out. If it is a very, I am speaking if it is a very superficial, okay. Okay, that aberrations, what will happen? There are chances of spread of infection. So this layer, the second layer, Bowman's membrane is acting as a barrier. So there are chances of infection spread if the wound is deeper. If it also cut through the Bowman's membrane, this protection is lost. Okay. Once damaged, this Bowman's membrane cannot regenerate. Okay, leading to scar formation. So what will happen if a scar is formed on the transparent cornea? There will be impairment of vision. Isn't it? The transparency will be lost in that portion of scar. So once damaged, the Bowman's membrane cannot regenerate. Because of scar formation, there is impairment of vision. So next layer, corneal stroma. Corneal stroma, that is C. Okay, third layer. Here, what is happening? What is the other name? It is substantia propria, and it forms the most of the thickness of cornea. So whenever you draw, the thickest layer that you should show is the corneal stroma. Okay, it is made up of type 1 collagen fibers in ground substance, and they contain sulfated glycosaminoglycans arranged with great regularity and form lamellae. Okay, about 200 to 250 layers of lamellae are arranged. Fibers in one lamellae are parallel to one another, but those in the adjoining lamellae run in different directions, forming obtuse angles with each other. Now, the transparency of cornea, we have already mentioned. First of all, we have mentioned it is because the cornea is avascular. Yes. Another cause of transparency, it is due to this regular arrangement of fibers and also due to the fact that the fibers and the ground substance have the same refractive index. Okay, so there is no refraction of light rays. They will go through a same line because the refractive index of the two media are same. Okay, then fibroblast, what happens? I mean, within the cornea, okay. So, because of the arrangement of fiber, there is no scattering because the refractive index is same. Then fibroblast, they are the cells, okay. They are also known as keratocytes or corneal corpuscles. They appear to be flattened in vertical sections through the cornea, but are seen to be star-shaped on the surface view. Then the fourth layer is for B, okay, Descemet's membrane. This is the posterior limiting lamina. I already mentioned Bowman's membrane was anterior limiting lamina. So the posterior limiting lamina is the Descemet's membrane. This is homogeneous and a cellular layer, okay. This is the true basement membrane. This is considered as a true basement membrane on which the cornea rests. They can regenerate after injury. So which was the layer that cannot regenerate after the injury? The second layer, the anterior limiting lamina, okay, or the Bowman's membrane was not regenerating after injury, but Descemet's membrane can regenerate after injury. At the margin of the cornea, the posterior limiting membrane becomes continuous with the fibers. It forms a network in the iridocorneal. What do you mean by iridocorneal angle? That is a junction between the iris and the cornea. Okay. So, the innermost layer, it is for the fifth layer, that is my E. What is that? Endothelium. So, which will be lining the posterior surface of the cornea and endothelium. This is a single layer of flattened cell. Okay. Anterior layer, it was non-keratinized stratified squamous. There were several layers for this. There was superficial flattened layer, middle polygonal, deeper columnosus. But here, it is a single layer of flattened cell that constitutes the endothelium. So, it will be lining what? Which chamber? Cornea comes in front of the anterior chamber. We are speaking specifically about cornea. So, it will be lining the anterior part of the anterior chamber. Okay. And it will come in contact with aqueous humor, obviously. So, what will happen? The endothelial cells, they possess numerous mitochondria and are adapted for ion transportation. So, they are united to the neighboring cells by desmosomes and by occluding junctions. Then cornea have to be maintained its transparency. So the excessive fluid from the cornea is being pumped out with the help of this endothelial cells. So already we have discussed the causes of corneal transmission. Some of them, first of all, avascularity, regular arrangement of fibers in the substantia propria, same refractive index of the collagen fibers and the ground substance, regular and uniform arrangement of the anterior epithelium. Okay, then relative state of, because of this pumping out of excessive fluid here, it is maintaining transparency. That is a relative state of corneal dehydration. Okay, so but it should not be dehydrated that is why there is anteriorly there is a tear film, posteriorly there is aqueous humor and all. But when you consider about the thickness of the, throughout the thickness of the cornea, it is relatively in a dehydrated state. So we have mentioned that it is avascular. So where it will get the nutrition from? Okay, the nutrition of the cornea is from the lacrimal fluid forming thin capillary layer. That capillary layer means, you know, the capillary rise, thin membrane fluid. That is what you mentioned here as capillary layer. That is the lacrimal fluid forming thin capillary layer. Aqueous humor in the anterior chamber, and we have mentioned about the sclerocorneal junction. There are blood vessels at the sclerocorneal junction. Okay, this is how you draw the cornea. So I have mentioned the thickest layer. What is the thickest layer here? What is the thickest layer I have shown? Which part? What is this? Someone answer?
Corneal stroma.
Yeah. So when you draw, you should be drawing 3/4, more than 3/4 of the diagram will be corneal stroma, isn't it? So the inner lining is a single layer of cell. What is that? Corneal endothelium. It is resting on the true basement membrane or the posterior limiting lamina. Can you see that line here? Okay, that is nothing but the posterior limiting membrane or the, what we mentioned as the, it is the actual basement membrane. What is that? The Descemet's membrane. So we have considered the 3 layers from posterior. Okay, this is the anterior, this is the posterior. Endothelium, Descemet's membrane, corneal stroma we have mentioned. So what will be that? This part, 2 more layers are there. What type of epithelium is this? This thickness, we have already learned to draw this epithelium. What is that?
Non-keratinized stratified squamous.
Yeah, it is written here, non-keratinized stratified squamous epithelium. So that forms the corneal epithelium. You can see columnar cells, polygonal cells, 2 layers, 2 to 3 layers, and the uppermost layer by. It should not be as thick as you draw your skin or so, because it is itself is a thin structure, right? So this epithelium should be drawn small, but you should show the 3 layers. Okay, then you should show the thickest layer here. So in between this epithelium and the corneal stroma, there is a, as we have drawn a line here, there is an anterior limiting lamina. That is the membrane, basically like a basement membrane on which this epithelium rests. That is the Bowman's membrane. So have you understand how to draw? So you can see some things here. What are they? They are nothing but the nucleus of the fibroblast in the corneal stroma. Okay, these blue lines. Okay, they are the nucleus of fibroblast. So this is how you draw the cornea. It is easy if you learn the layers correctly. Okay. Okay. Now about the next quote. So sometimes we'll be running short of time. So I'll be going to retina now. After we complete retina, we shall discuss these parts. Okay. Is there any problem? Is it okay for you if I discuss retina first? Yes. Because this is what you need. Okay. Retina is very important for you. So we shall discuss retina first and then come back if time permits to uvea. Okay. So about the retina. So how many layers were there for cornea? Someone please answer first.
5.
5 layers.
Okay. Cornea is a continuation of sclera. Okay. And it has got, cornea has got 5 layers. Okay. What was the middle layer? What is the middle layer? Not of the
Corneal
cornea. Of
stroma.
the eyeball. I mean of the eyeball as a whole. What is the middle layer?
Choroid.
Choroid. That is the vascular layer. So that will be rich of blood supply. And what is the innermost layer?
Retina.
Retina. What is the type of? It is the nervous coat. Okay. It's a nervous coat of the eyeball. Actually, the sense of vision is mainly taking part because of this layer only. This is the inner coat of the eyeball and lines the posterior 3/4 of the surface of the eyeball, inner surface. When I say about the inner surface, let me ask you, is it coming in contact with the vitreous humor? I mean, in contact with the posterior, in contact with the vitreous humor or not?
Yes.
Yes. So when we consider the eyeball, it is like a closed cavity. I mean, it is like a ball, right? So the inner lining is the retina. Don't get confused when I mention the inner. You should not be thinking when we look at the anatomy, the posterior pole, the out, മനസ്സിലായോ ബോളിന്റെ പുറകു വശത്തല്ല the what you call retina. Okay, it is the inner lining of the eyeball. Okay, and lines the posterior 3/4 of surface. It contains rods and cones. They are nothing but the photoreceptors. So I have already mentioned there are specialized area where vision is most acute. You can see in this diagram, this is the fovea centralis or the macula, which contains only cones, which are essentially bare. The overlying layers, that is, all the other layers have been pushed to the side in this area of fovea. So there is more only cones. Okay. Then what are the other things that we can see in the retina? There is something medially. Actually, this is medial, this is lateral. Okay, consider it as that. So medially, there is all these nerve fibers will converge and emerge as the optic nerve. So that portion, there will be no cones. There will be, if the light falls in this area, you can't see. That is a blind spot you mentioned. Okay, that is the portion of the optic disc where the optic nerve leaves the eye and there are no photoreceptor in this area. Okay, this portion. Whereas this portion, there is maximum photoreceptors. What about the rods and cones? Rods are specialized to perceive dim light, while cone cells perceive bright light and color. Okay, and these photoreceptor cells transfer the light energy into receptor potential. So there is transfer of energy, light to receptor potential. So this is where you see. Okay, so this is your fovea. There is the macula lutea. There is a depression, and the depression of the fovea is known as the fovea centralis is known as the foveola, which is the area of the clearest vision. Okay, medial to this is the optic nervous emerging. Okay, this is the posterior pole of the eyeball. Medial to the posterior pole, it is the optic nerve emerging or the blind spot. We have already discussed this. Okay. The retina can be said to have an external surface. Okay, when we are talking about retina specifically. So this is the posterior 3/4 of the eyeball covered by the retina from inside. So this retina itself is a layer, okay, that is having an external surface that is in contact with what? Contact with this pink layer, that is the choroid, and an internal surface that is in contact with what? Vitreous. And this thing, this yellow part, okay, consider it as yellow. Okay, this thin part itself is having 10 layers. Okay, cornea, we have mentioned 5 layers, but for retina, there is 10 layers, and these are all neuronal cells, okay, and they contain what? They contain processes. So these cell bodies, processes, all these things are arranged in several layers. You can here see a layer of cell body. Then you can see a layer of synapses between this cell and this cell. Then you can see the axons of this cell. This is a neuron. Consider this as a neuron. So this is axon. This can be dendrites. Okay. So there is synapse between these cells and this cell. So this mix in one layer. Then you have the cell body of this neurons. Then you have synapse between this type of neurons and this type of neuron. So synapses, cell bodies, all these things are arranged in different layers, and that all makes towards 10 layers of retina. So when we consider the light is entering from the anterior part, from this part. So light will be falling onto this surface. What is the surface I mentioned? Is this the external or the internal surface of the cornea? That is in contact with the vitreous, that is the internal surface. So light will be falling onto the internal surface. So this part coming in contact with the choroid is the external surface. So this portion will be more towards the internal surface, and this portion here will be the external surface. So what I mentioned in the external surface? In contact with the choroid, there is the pigment epithelium. Then comes the rods and cones. From here to here, I mentioned. Then there are many layers. We will look into that layers. So what are the layers of the retina? First of all, the retinal pigment epithelium. Okay. Then comes the layers of rods and cones. You have mentioned the rods and cones are photoreceptor cells. Then there is a layer of external limiting membrane. You can see here. Okay. Then outer nuclear layer. So let me discuss this. So this is one photoreceptor cell. This is another photoreceptor cell. So here they are mentioned. This is the rods. This is the cone. How will you differentiate? Look at the end. The end of this rod cells is rod-like, where here it is a conical projection. That is why I mentioned rods for this and cones for this. Okay. So the processes of this layer make processes of this is rods and cones, processes of the rods and cone photoreceptor cell. This is from here to here, it is photoreceptors. Okay. This layer, photoreceptor layer, is made by the processes of these cells. Then what about the another layer? Here we can see the cell bodies of all these photoreceptors. Okay. Before that, there is the external limiting membrane. The cell bodies of all these rods and cones is forming the outer nuclear layer. Then what is the next layer? Outer plexiform layer. What is that? These rods and cone cells are synapsing with the neurons from the next layer. Neurons of the inner nuclear layer are synapsing with the photoreceptor cell at this layer. Can you see the synapse here? So that is the plexiform. So plexiform layer, you think there is synapse. So outer plexiform is synapse between the cells of inner nuclear layer and cells of outer nuclear layer. Then comes the inner nuclear layer. Inner nuclear layer is having some other neuronal cells that we will learning now. you'll be learning. So cell bodies of that layer, neurons are arranged in the inner nuclear layer. Then inner plexiform layer. So what is plexiform? There is synapse. So there is, what is that? There is synapse between the cells of inner nuclear layer and cells from above layer. What is above layer? Ganglion cell layer. So have you understand something about the layers? So there is alternating the layer of cell body, there is synapse, there is cell body, there is synapse, like that. Here you can see cell body, synapse, cell body, synapse. That is, you mentioned outer nuclear, outer plexiform, inner nuclear, inner plexiform, and outermost ganglion. From there, what happened? The nerve fiber is emerging. Where will be going through? These nerve fibers from the ganglion cell layer will actually go and forms what? Optic nerve. Have you got a little idea about the layers? Is it clear for you?
Yes, ma'am.
Okay, then only I can move forward. You should be clear about this layers. Okay, so now we are discussing about the pigment cell layer. Pigment cell layer is nothing but the outermost layer of retina, separated from the choroid by the Bruch's membrane. Okay, it is consisted of a single layer of low cuboidal cells with basal nucleus containing melanin pigment. You can see nucleus and the melanin pigment. The process of the pigment cell extends into the next layer. Each pigment cell is related to dozens of rods. You can see rods and cones here. So this is a single pigment cell and each of them is related to dozens of rods and cones. Electron microscopy, we can see large microvilli that contains pigments. Okay, this is a large microvilli that has pigments. You can see brown spots in between. Pigments projecting into the intervals between the rods and cones, and the plasma membrane at the base of the cells show numerous infoldings. Okay, this is how you describe a pigment cell layer. So that was the first layer. What are the functions of the pigment cell layer? So the pigment cell layer will actually help in absorption of the scattered light. It provides the blood retinal barrier function that we will discuss in later. Then helps visual pigment regeneration and synthesis is taking place in this layer. There is synthesis of growth factors. There is maintenance of retinal adhesion. Retina have to be adhered to the underlying layer, it's the choroid. That is by the pigment cell layer. Okay. Phagocytosis. What phagocytosis? It is actually phagocytosing the older tips of this rods and cones. Okay, it is regenerating. Okay, these tips of the rods and cones are being phagocytosed by the pigment cells. Okay, then electrical homeostasis and repair and regeneration after injury. So what is your second layer? Second layer over here you can see what is, if it was the pigment cell layer, first layer, second layer will be the processes of rods and cones. This is the process of rods and cones. Okay, that is the second layer. Rods are processes of rod cells and cones are processes of cone cells. So you can see these are the processes. Okay, this layer consists of the outer segment, that is the peripheral process of rods and cones, and it is having the photoreceptor function. Okay, when we consider the rods and cones, you have to know about the ultrastructure of rods and cones. So this is how you look at the rod and cone. Both of them are looking almost similar in structure, and they convert the stimulus of light into nerve impulse. Okay, each rod cell or cone cells can be regarded as a modified neuron. It consists of a cell body. You can see both of them. A peripheral process and a central process. So this external part is the peripheral process, internal part is the central process. The peripheral process lie in the layer of rod and cone as described. This peripheral process is lying in the layer of rods and cones. So nuclei of the cells are arranged in several layers in the form of the outer nuclear or the external nuclear that I have mentioned earlier. The nuclear cell body of these photoreceptors are arranged in the third layer, that is the what we call the outer nuclear layer. So the outer nuclear layer will be darkly stained because there is nucleus there. The central process, that is the inner process. The central process of each rod cell or cone cell is an axon. So that will be synapsing with the nucleus from the above layer. So it extends into the external plexiform layer where it synapses with the dendrites of the bipolar cell. Hope you understood the parts of the rods and cones. So what is the difference between the rod and cone that you have to know now? Okay. Before that, how many rods and cones will be there? There are about 7 million cones in each retina. Each retina means in one eye. Okay. And rods are more in number. There are more than 100 million rods. Okay. What is the cones helpful for? They are helpful for bright light. Okay. Photopic vision. Whereas they can discriminate color. Okay. Rods, what about rods? They are responding to poor light or scotopic vision. And especially for the movement across the field of vision, we can appreciate because of the presence of this rods. Okay, what is the pigments? Pigment in the rod is rhodopsin, whereas the pigment in the cone is iodopsin. Okay, now considering together almost similar, but can you see the lower end? The lower end is different, isn't it? So about the rod, it is known as spherule, whereas for the cone, it is known as the pedicle. Okay. This is about the ultrastructure in a short. Then what is the next layer? Next layer I have mentioned. Second layer was layer of rods and cones that contains the outer segments or the peripheral processes of rods and cones. Then comes the next layer. What will be that? What will be the third layer? That will be the cell bodies of rods and cones. Okay. What will be known as? It is the external nuclear layer. Between the external nuclear layer, that is actually external nuclear layer is the fourth layer. Before that, there is a membrane known as the outer limiting membrane or lamina. A presence of pink linear marking called as outer limiting membrane or lamina, resulting due to the Zoniga adherence of the glial cells that or Muller cells with the photoreceptor cell. They are supporting cells, okay, and is radially oriented in the retina. That cells forms the outer limiting membrane. And deeper to that, we have the external nuclear layer, as we have mentioned. That is what we are seeing here, the cell bodies of the nuclei of rods and cones. Then what is the fifth layer? That will be actually the synapse between the, we have already mentioned, spicules, spherules. Okay. So there will be synapse between that portion of the rods and cones and with the above cell. What is the above cell? Above cells is nothing but the bipolar cells of the next layer. Okay. So the external plexiform layer, also known as the outer synaptic zone, consists of nerve fibers and forms a plexus. Axons of the rods and cones synapse here with the dendrites of the bipolar neurons, and there are some horizontal cells here. Okay, all these synapses here. Okay, this layer stains slightly. There is no nucleus, so it stains slightly. Whereas I mentioned this layer will stain darkly because of nucleus. So what about the next layer? That will be the internal nuclear layer. So the cell bodies of these bipolar cells will be arranged in the next layer of the internal nuclear layer. So the cells there are bipolar cell. What is mean by bipolar? That is having a dendrite which enter exit external plexiform layer, then axons which will enter internal plexiform layer. Okay, this is the dendrites entering here and axons entering here. That is about the bipolar cells. Okay, they are arranged perpendicular to the layers of retina. Layers of retina like this, but the cells are arranged perpendicular to the layers of retina. Then comes the horizontal cell. We have mentioned horizontal cells here. What about them? They give the processes that run parallel to the retina surface, which enters the outer plexiform layer and synapse with rods, cones, and dendrites of bipolar. Here you can see the horizontal cell. They are actually arranged parallel to the layers of retina, and their dendrites can be seen synapsing with rods, cones, bipolar. Okay, this is about the horizontal cell. So another cells present, amacrine cells, Muller cells. What are they? Amacrine cells are also lying horizontally in the retina, and their processes enter the inner plexiform layer where they synapse with the axons of bipolar cells. Okay, they are having no true axons. Okay, processes of the amacrine cells will conduct impulse in both directions because they are horizontal cells. What is their function? They will be having interaction between the adjacent area of retina, resulting in production of sharp images. Okay. They also involved in the analysis of motion in the field of vision. That is about amacrine cell. Next, Muller cell. All now we have mentioned are, what were they? Neuronal cells. Muller cells are nothing but retinal gliocytes with numerous protoplasmic processes. that will extend almost through the whole thickness of the retina. Externally, they extend to the junction of layer of rods and cones with the external nuclear layer. By the process of adjoining gliocytes meet to form a thin external limiting membrane. Internally, the gliocytes extend to the internal surface of the retina and forms the internal limiting membrane. Already we have mentioned external limiting, and coming we will be mentioning internal limiting. These are actually formed by the Muller cells. So they are nothing but the retinal gliocytes. What will they function? They will help in supporting the neurons of the retina and ensheaths them. What is the next layer, the seventh layer? That will be the plexiform layer, inner plexiform layer. What will be happening? There will be synapses between the axons of bipolar cells and the dendrites of the above layer, that is the dendrites of ganglion cells. Okay, they will synapse with each other and also with the process of amacrine cell. Then next layer, ganglion cells. So what will be there? There will be cell bodies of ganglion cells. The dendrites will enter IPL. IPL means internal plexiform layer to synapse with the bipolar cells. Okay, and what will happen? It will, outer fibers will form the ninth layer. That is the layer of optic nerve fibers. That is formed by which cell? Ganglion cells in the fibers. Okay, axons of the ganglion cell. The fibers converge on the which part? All the fibers will converge on the optic disc. I have already mentioned, where they pass to the foramina of the lamina cribrosa. Do you remember lamina cribrosa? See, like things in the sclera. So they will pass through them and they will form the optic nerve. Okay, then what about internal limiting membrane? Already mentioned, limiting membrane will be formed by what type of cell? Muller cell. Okay, the process of the adjacent Muller cell, they are supporting cells. Okay, extends to the internal surface of the retina to form the internal limiting membrane. They separates the retina from the vitreous. So this will be very, you may know there are 10 layers to remember or study, but it's actually easy. If you consider the cells in each layer, you can study the photoreceptor cells, you can study the bipolar cell, you can study the ganglion cell. They are arranged one above the other. And lower process will make one layer for this. Then cell bodies, then synapse, then cell bodies, then synapse, then cell bodies, then nerve fibers. Actually, this is easy. Okay, don't get panic if there are 10 layers to study. If you study the cells first and then consider they are arranged one above the other, you can easily make out the 10 layers. This is about the 10 layers of the retina. So this is how the retina will appear in H&E staining. I am not showing you the demonstration slides here because you will be seeing it in the lab itself. Just for your reference, this is the light-centric. So this is the inner part. So this is the choroid. So this is the outer part. So there is a retinal pigment layer. This is the layer of rods and cones. There is the outer nuclear layer and the inner nuclear. Can you see the nuclear layers are very well stained? Okay, the inner and the outer nuclear layer can be made out even at low magnification because they are having the nucleus. Outer nuclear is thicker, isn't it? Outer nuclear is thicker and the nuclei are more densely packed than the inner nuclear layer. Okay, then nuclear in the inner layer belongs to bipolar cells. What about this? To the rods and cones. Okay, then ganglion cell layer. Can you see the ganglion cell layer? This is actually a layer of nucleons, but it is made up of almost a single row, not that much crowded nucleus. Almost a single row of nucleus that is having what? Varying size. Okay, they will be having an indistinct cell outline, but the nuclei can be made out. They are of various size. Okay. They will be larger nuclei, but they will be staining more likely than the nuclei in the inner and the outer layer. These all things you should make note while you draw. Okay. Then comes what? The layer of, here you have the layer of pigment cells. Okay. The layer of pigment cells, they will resemble a small low cuboidal epithelium, and all the nuclei in this layer are of similar size. The remaining layers, the layer of rods and cones here. Can you make it out clearly? The outer plexiform layer. All these layers are seen as light staining areas. We cannot make much details in this area using H&E staining. Okay. So the layer of rods and cones can show some vertical striations. This is all the processes of the rods and cones. This is how we can make out the H&E, under H&E staining, you can make out the retina. So another important thing is about the blood retinal barrier. So blood vessels that ramify in the retina do not supply the rods and cones. They are supplied by diffusion from the choroidal vessels. Endothelial cells of capillaries in the retina are united by tight junctions to prevent diffusion of substances into the rods and cones. This is referred to as blood retinal barrier. So this is how you draw the retina. Okay, so I'll be uploading these diagrams in the Google Classroom. Yeah, now your time is up. Actually, I have mentioned all the point that is very important for you, retina and cornea. The remaining portion, lens, uvea, and the eyelids. I mentioned eyelids is a new core competency part, but you are having yourself a seminar for that portion. So go through that. If any doubt, you can ask. Okay. Now your time is up. Actually, we have covered the important parts. Any doubts? Any doubts for you? Anyone? Anyone please postpone? We can wind up the class.
No doubts, ma'am.
Okay, thank you.
Thank you, ma'am.
Thank you, ma'am.
Thank you, ma'am.
Thank you, ma'am.
Okay.
Good morning, all.
Good morning, ma'am.
Okay. Can you all see the slide?
Yes, ma'am.
Okay. Only 100 can join. Is it like that? Only 86?
Yes, ma'am.
Okay. The others are watching through YouTube.
Yes, ma'am.
Okay. Okay. So in the last class, regarding the heme metabolism, we were discussing about what were we discussing? Anyone? We have finished the heme catabolism and we have seen the disorders associated with the heme catabolism, which is called jaundice. Okay, different types of jaundice we have seen. In the last class, we discussed about the congenital and acquired causes of jaundice, like Dubin Johnson, Rotter, Krigler, Nudger, Gilbert, etc. And some other acquired causes like congenital spherocytosis, glucose 6-phosphate deficiency, autoimmune hemolytic anemia, toxins like carbon tetrachloride. All these can cause hemolytic disease of adults. And jaundice, we can classify into 3 types. Which are those 3 types?
Prehepatic.
Prehepatic or hemolytic jaundice. Then hepatocellular jaundice and
Obstructive jaundice.
posthepatic or obstructive jaundice. Okay. So this hemolytic disease of adults is a prehepatic jaundice where the defect is before the conjugation, means there is increased breakdown of RBC which is occurring in the reticuloendothelial system, resulting in formation of huge amount of unconjugated bilirubin. The liver cannot handle that much amount of bilirubin, so only normal amount of bilirubin can only be conjugated by the conjugating enzyme UDP, glucuronide transferase. So the rest of the thing will be there in the blood as unconjugated bilirubin. So the clinical feature is due to increased amount of unconjugated bilirubin in the blood. Okay, and total bilirubin concentration in hemolytic disease, it will be less than 4 milligram per deciliter. And coming to the second kind of jaundice, which is a hepatocellular jaundice, which means there is a hepatocellular damage. Hepatic cells are damaged. So it is the hepatic cells that is conjugating. The conjugating enzyme is located inside the hepatic cell. So when there is an injury, toxic damage to the hepatocytes, the conjugation will be affected. So the unconjugated bilirubin will be able to reach inside the hepatocyte, but conjugation will be defective or reduced. Some of the hepatocytes will be functioning, not all hepatocytes will be damaged. So some of the conjugation will be occurring, but mostly the other cells, the damaged cells, will not be able to conjugate the bilirubin. The causes, there are a long list of causes for hepatocellular jaundice. All the damage, all the causes which cause damage to the hepatocytes can result in hepatocellular jaundice. Of this, the most common cause is the viral hepatitis. Viral hepatitis, there are A, B, C, different varieties. Hepatitis B is more common. And alcohol, the alcohol can cause alcoholic liver disease. It can damage the liver. It can result in hepatocellular jaundice. There are different toxins like carbon tetrachloride. All these can damage the hepatocytes, which can result in hepatocellular jaundice. And there are certain immunological causes there, like antibodies are directed against our own cells, and thus the hepatocytes may be damaged, like autoimmune, that is autoimmunity. The reaction, antibody reaction against our own cells, that can result in the hepatocyte damage, resulting in hepatocellular jaundice. And NASH is non-alcoholic steatohepatitis. Non-alcoholic steatohepatitis. We will study the details in lipid metabolism. And there are certain general diseases like Wilson's disease, hemochromatosis, porphyria. In all these, the liver will be damaged and can result in jaundice. And some cancers, especially the cancers of the liver, hepatocellular carcinoma, or mostly the metastasis, means the spread of the cancer can occur to liver, bone, brain, etc. So when a metastasis occurs to the liver will get damaged or will not be able to function properly. In that case, also jaundice will be there. Lymphoma is a cancer, increased proliferation of the lymphoid tissue. Okay, it can also result in lymph nodes will be enlarged, can also cause hepatocellular jaundice. Some infections like TB, leptospirosis also cause hepatocellular jaundice. Parasitic infection, drugs like salicylate, methotrexate, rifampicin, it is a drug used for some cancers. Rifampicin is a drug used for the TB. All this can cause liver cell damage that can result in hepatocellular jaundice. I told you the most common cause is the viral hepatitis. There is a difference. Initially, there will be unconjugated hyperbilirubinemia, which means it is a damage to the liver cell. It is a liver cell that is conjugating. So conjugation is somewhat defective. Yes, but some of the conjugation will be functioning. If it is getting conjugated, some of the liver cells functioning, they will conjugate. That conjugated one will be able to reach the intestine. Okay, the rest of the things will be normal. Okay, so initially there will be unconjugated hyperbilirubinemia, but in the second stage of this hepatocellular jaundice, there occurs another thing. So see, these are the hepatocytes. The red ones are the hepatocytes. In between that, we can see the biliary canaliculae, bile canaliculae. So after conjugation, the conjugated bilirubin is sent to this biliary canaliculae through some protein MRP2 or MOAT. Okay, you remember? Yes or no? I've discussed.
Yes,
it. Okay, so in the first stage, it is the problem with the hepatocytes. Some of the hepatocytes will be conjugating. That conjugated one can easily reach the biliary canaliculae and it can reach the intestine. So there won't be any conjugated bilirubin increased in the blood. but unconjugated will be increased because large amount of unconjugated bilirubin are there, but only few can be conjugated. So initial phase it is unconjugated hyperbilirubinemia. Okay, but in the later stage, due to this liver cell damage or the infection, the liver cells or the hepatocytes may get enlarged. They become edematous. So this one will enlarge like this. This one will also be enlarged like this. So what happens? The biliary canaliculae will be obliterated. There will be a block to the biliary canaliculae due to this edema of the hepatocytes. So what happens is that even though some of the conjugated bilirubin are formed within the hepatocytes, it is due to this edema and the block to the flow of the unconjugated bilirubin to the biliary, conjugated bilirubin to the biliary canaliculae. What happens? Some conjugated bilirubin will also be there in the hepatocyte. They will come back to the blood. So in the later phase of the hepatocellular jaundice, there will be both unconjugated and conjugated bilirubin in the blood. Is it clear for you? Yes or no? So initial phase, it is the problem with the conjugation because the liver cell is damaged, but some of the conjugation will be occurring. The conjugated one will be able to reach the biliary canaliculae. So in the blood, only unconjugated bilirubin will be there. In the later stage, due to the disease, infection or the some damage, the liver cell or the hepatocytes will be enlarged or become edematous. Due to edema, they will cause a block to the biliary canaliculae. So even though some conjugated bilirubin are formed, it cannot be emptied into the biliary canaliculae, resulting in formation of the conjugated bilirubin, retaining the conjugated bilirubin in the blood. So in the later stage, both conjugated and unconjugated bilirubin will be present in the blood. What happens in the urine? In your urine, you know, there is unconjugated hyperbilirubinemia, and the second phase, both conjugated and unconjugated bilirubin. What happens to this conjugated? The conjugated bilirubin can be able to, it is soluble in blood, right? Soluble in urine also. Okay. So the conjugated one, when present, it can be excreted through the urine. So bilirubin will be there in the urine. The Fauci's test will be positive because some conjugated bilirubin is there in the later stage, and that can be excreted through the urine. Okay. And coming to the last type of jaundice, that is obstructive jaundice. It can be due to obstruction anywhere in the pathway from the biliary canaliculae to the part where it reaches the intestine. Okay. Anywhere in the pathway of the flow of the bile, if there is an obstruction, it can result in obstructive jaundice. We can classify this obstructive jaundice into two types. It can be intrahepatic cholestasis. Cholestasis means it is an obstruction to the bile flow. Okay. So intrahepatic means somewhere inside the liver. Within the liver itself, if there is obstruction, it can result in intrahepatic cholestasis. Somewhere outside the liver, up to the reaching the point, in the reach point in the intestine, if there is obstruction, it can result in extrahepatic cholestasis. Let's see the causes of the intrahepatic cholestasis. Chronic active hepatitis can result in a obstruction. Okay. I told you in the later stage, there is an obstruction due to the edema, the bile flow will be obstruction. Okay. So that can result in a cholestasis or the obstructive jaundice. And biliary cirrhosis. Okay. The biliary part, if there is the cirrhosis affecting the biliary part, it can result in biliary cirrhosis. Lymphoma, when there are enlarged lymph nodes as a part of carcinoma, this enlarged lymph nodes within the liver, that can result in obstruction, intrahepatic obstruction, or it can be a liver carcinoma like primary hepatoma can result in the obstructive jaundice. Extrahepatic cholestasis, the causes is commonest is gallstones. When there are gallstones present in the gallbladder, they come and obstruct the, so this is the gallbladder. If there are some gallstones, it can come and obstruct this mouth. If two of them come and obstruct this mouth, the bile cannot be flow into the biliary tract and finally reaching the intestine. Okay. So extrahepatic cholestasis, the cause is gallstones, then carcinoma head of pancreas. In the anatomy, you might have studied the structure and the position of this pancreas. The pancreas is very near to this liver. And when there is a cancer or the carcinoma to the head of the pancreas, the pancreatic cells, they may enlarge, hypertrophied, and they come and press onto the biliary part. Okay, to the bile, there will be an obstruction. So extrahepatic obstruction will be there by the carcinoma head of pancreas, and in the anywhere in the porta hepatis, if there is an enlarged lymph node, that can also press onto the biliary tract and can cause obstruction. I hope you are clear about the different causes of obstructive jaundice. You should know there are two classifications to extrahepatic cholestasis and intrahepatic, and you should know some 2 or 3 causes of each cholestasis. Okay, shall I move on?
Yes, ma'am.
Okay. Then we will see, you have to answer which all things are increased in all these conditions. So when asked about the investigation, there are 3 kinds of investigation. The parameters which are increased or decreased, altered in blood, altered in urine, and altered in feces. So you have to specifically make it as 3 headings: blood findings, urine findings, and findings in the stools. Okay. So we'll see the 3 causes: hemolytic, hepatocellular, and obstructive jaundice. In all these types, which all things are increased. So first is unconjugated bilirubin. What will be increased in hemolytic jaundice? Unconjugated, increased, decreased, or normal? You have to answer.
Increased.
Increased. Why? Because it is due to lysis, hemolysis. The RBC will be lysed. So after the lysis, they will be catabolized to form the unconjugated bilirubin. So it will be increased. What happens in hepatocellular jaundice? Unconjugated bilirubin.
Increased.
It will be increased. What happens in obstruction?
Normal.
Okay, it will be normal because up to conjugation it is normal, but it is due to the obstruction to the bile flow. So it will be normal. What will be the findings of this conjugated bilirubin in the hemolytic jaundice?
Normal.
Okay, it will be normal. In the case of hepatocellular jaundice, it will be increased, right? In the second phase, I told you there will be an obstruction. So even though conjugated bilirubin is formed, it cannot be emptied into the biliary canaliculi. So it will be increased. You got the concept?
Yes.
Yes,
Okay.
ma'am.
What happens in obstructive jaundice?
Increased.
Increased. Okay. And you have to say one more thing, ALP. I think it was covered in enzymology. What is ALP? It's an enzyme marker for
liver disease.
Alkaline phosphatase.
alkaline phosphatase is a marker for hepatocellular damage or obstruction.
Obstruction.
It is a specific marker for obstruction. So will it be increased in hemolytic jaundice?
No.
No. What happens in hepatocellular jaundice? Increased, decreased, or normal?
Normal.
I told you in the second phase of hepatocellular jaundice, there is an obstruction due to the edema of the hepatocyte. So it is somewhat increased. See what happens in obstruction, it is very much increased. Actually, it is here in this obstruction, there is a complete obstruction causing 10 to 12 times increase on this enzyme. But in the hepatocellular jaundice, in the second phase, due to the edema of the hepatocyte, there is some obstruction causing somewhat increase, a moderate increase of the alkaline phosphatase. Coming to the urinary findings, what will be the things which we look in urine? എന്തൊക്കെയാണ്?
Conjugated bilirubin, urobilinogen.
Okay. Bile salts, bile pigments, and urobilinogen will be checked. The bile salts will be same as like that of the bile pigment. Bilirubin is the bile pigment. Bilirubin and biliverdins are the bile pigment. Along with this bile pigment, bile salts are also excreted through the bile. So both can be looked for. The findings will be almost same. Okay. In hemolytic jaundice, what happens to the bilirubin? Will it be present? It will be unconjugated, so it cannot be excreted, so it will be absent. So like bile salts, like bile pigment, bile salts, so it will be absent. What happens in hepatocellular jaundice? Here also mainly it is unconjugated, so it is absent. What happens in obstruction? Here it is the conjugated bilirubin, so it can be excreted, so it will be present, same as bilirubin. So this is a difference is that I told you in the second phase there is an obstruction, that is why it is written as present. Okay, when you write it as present, you have to just mention in a bracket. Okay, in the second phase there is an obstruction, it can result in excretion of a small amount of bile salt and bilirubin in the urine. What happens to the urobilinogen? Will it be normally present in urine? Specifically, I have told in the last class. Will it be normally present? How much?
In very
Small.
small quantity.
Very small quantity. Normally, it is present only in trace amount. Less than 4 milligram per deciliter is normally present in urine. And what happens in hemolysis? What happens? It will be?
Increased.
It will be increased. What happens in hepatocellular jaundice? So there are two phases. Initially, there is unconjugated bilirubinemia. In unconjugated, it will be increased. In the obstruction, will it be increased? If there is an obstruction, the conjugated bilirubin cannot reach intestine. When it reaches intestine, only it is deconjugated, and by the action of the bacterial enzyme, it is converted to urobilinogen. So if there is obstruction, it is unable to reach the intestine. As it is not coming to the intestine, it is not converted to the urobilinogen. So urobilinogen will not be there or somewhat absent or decreased in the obstructive phase, but it is increased in the early phase. Early phase, it is unconjugated hyperbilirubinemia. Is it clear for you?
Yes, ma'am.
Okay.
Yes,
What
ma'am.
happens in obstruction? Obstructive jaundice?
Absent.
It will be absent. Good. What happens to the feces? What will be the thing which we look for?
Stercobilin.
Urobilins and stercobilins. What will be the thing in hemolytic jaundice? It is somewhat same like the urobilinogen. This will be normal or decreased. In the initial phase, it is unconjugated bilirubinemia. In that case, it can be normal or it can be decreased in the obstructive phase. In the case of obstructive jaundice, as there is obstruction, it is not reaching. The urobilinogen is not formed in the intestine. From the urobilinogen, only the stercobilinogen and stercobilin and urobilin are formed. As it is not formed, the color of the stools will be clay colored. Urobilins will be absent. That is why the normal color of the stool is due to the presence of, it is due to the presence of stercobilin and urobilin. Okay. So as they are absent, this golden yellow colors of the stools are not there. So clay colored stools will be seen. This is all about the jaundice, different types of jaundice and their findings. These table is very important for you. You have to study the findings, the blood findings, urine findings, fecal findings. All these in 3 types of jaundices. Okay. And we will move on to the next topic. Okay. So regarding the hemoglobin, can you all see the slide?
Yes, ma'am.
We'll move on to the next chapter, which is hemoglobin. Okay. Of this, the majority it was already covered in the physiology topics that won't be discussed here, because the transport of oxygen, transport of carbon dioxide, all these was discussed in the physiology classes, right? Oxygen dissociation curve, was it covered? The factors affecting the oxygen dissociation curve. Okay, so you go and read up. It may be asked for you, but we won't be discussing as it is already covered. Regarding the hemoglobin, when I talked about the heme synthesis, I've discussed about the structure of the heme. So there is no point in discussing it again and again. So it is a tetrameric protein and it is a globular in shape. Normal levels of hemoglobin is 14 to 16 in males and 13 to 15. Its molecular weight is 67,000. Adult hemoglobin has got this much pattern. The majority is made up of HbA. HbA means the globin chain difference is 2 alpha, alpha 2 and beta 2. What happens to HbA2? Alpha 2?
Gamma.
What happens to HbF? HbF?
Alpha 2 and delta 2.
Okay, alpha 2 and delta. Okay, normally only 1% of HbF is seen in an adult. So when at birth this HbF concentration will be high, it will be more percentage, but they will be hemolytes. There will be accelerated hemolysis. I told you in newborns there will be physiological jaundice due to conversion of this HbF to the HbA. So adults have only 1% of HbF and 2% of HbA. So when in diseases like thalassemias, etc., this HbA2 and HbF levels will be increased. We'll study what is thalassemias, what are sickle cell anemia, hemoglobinopathies, etc. So adult pattern is this: 97% is made of HbA. This is the normal structure of a hemoglobin. It has got 4 heme groups. This one is a heme. So there will be 4 hemes. The 4 heme will have got a central ion and a porphyrin ring. To that 4 groups, what is there? 2 alpha. This is an adult HbA. 2 alpha and 2 beta. You can see this is a 1 alpha chain, the other alpha chain, the other beta chain, the other one. Okay, this will be the structure of the hemoglobin. And the heme is called the prosthetic group, which means the non-protein part is the prosthetic group and the protein part is the globin chain. There will be total of 4 chains: 2 alpha, 2 beta, 2 alpha, 2 gamma, and 2 alpha, 2 delta. Okay. And there are some moieties which are important. There are 30 histidine residues. It is due to the presence of histidine residues. Hemoglobin will act as a buffer. Okay, the buffering action of hemoglobin is due to the presence of this histidine residues. The important some histidine residues are, if you can remember, it is good. Otherwise, it's okay. 58th position of the alpha chain, that histidine is called distal histidine, and the 87th position of the alpha chain is proximal histidine. And the 4 subunits are held by some non-covalent bond, and each subunit, I told you, it contains a heme moiety. Heme means it is an iron plus a porphyrin ring. Porphyrin is made up of 4 pyrrole rings which are linked by methynyl bridges. And regarding the alpha chain, beta chain, etc., the alpha chain, which each chains are polypeptide chains, which means amino acids are linked together, linked by peptide bonds. In the case of alpha chain, there are 141 amino acids. So it is called as a protein, the polypeptide chain. 141 amino acids are there. In the case of beta, gamma, delta, all they have 146 amino acids. Okay. When we'll study about the diseases associated with this, we can say some, if there is a change in amino acid, if one amino acid from any of the amino acid from this 141 amino acid change, it can result in disease. We'll discuss this in the coming classes. Okay. Iron, I told you, it occupies the central position of the porphyrin ring. It is always in the ferrous state, means Fe2 plus state. Okay. The same. Regarding the oxygen dissociation curve, I will just say it is a sigmoid shaped curve. It is depending upon certain factors like pH, PO2, PCO2, 2,3-BPG, etc. Right? You remember?
Yes, ma'am.
Okay. So ODC of the hemoglobin will be in a sigmoid shape, whereas the ODC of the myoglobin will be in a hyperbolic state. We'll have a slide on myoglobin, I will tell you. So the myoglobin ODC will be hyperbolic. So the differences will between the two proteins, the hemoglobin and myoglobin, will discuss shortly. Okay. So this is the oxygen dissociation curve. It means the PO2, the oxygen saturation of the hemoglobin at different partial pressures of oxygen is called the oxygen dissociation curve. If the 2,3-BPG level increases and if there is acidity or if there is increase in pH, this is the normal oxygen dissociation curve. It will be shifted to the right. If the opposite are the condition, it will result in shift of the curve to the left. Okay, what is Haldane effect?
Chloride shift.
Chloride shift. What is Bohr effect? So according to different pH and PCO2, it is the change in this pH and PCO2 that facilitate the oxygenation of the hemoglobin in the lungs. In the lungs, the oxygen tension will be high, PO2 will be high, and the PCO2 will be low. So when high oxygen is present in the surrounding, the hemoglobin will take up this oxygen and will release the carbon dioxide. What happens in the tissues? In the tissues, the oxygen is low. It is the venous side. Okay. So the carbon dioxide will be high. So at that point, from the tissue, the carbon dioxide is taken by the hemoglobin. Carbon dioxide is taken by the hemoglobin and oxygen is delivered to the tissues. So that difference, it is the same hemoglobin that take up oxygen and taking up this carbon dioxide. Depends upon the pH and the PCO2. So the influence of pH and PCO2 to facilitate the oxygenation of the hemoglobin in lungs and deoxygenation at the tissue, that is called as Bohr effect. Okay. Hamburger effect is, you go and check. So to maintain that neutrality, the chloride will come back to the RBC. Okay, that is called this Hamburger effect. Effect of temperature, effect of 2,3-BPG is important. You have to go and check what happens in increased altitude, what happens in babies, etc. The babies have high affinity because this, why? High affinity for oxygen. You go and check about the differences in 2,3-BPG. Okay, high oxygen affinity of the HbF. They have, the fetus and the newborns have increased amount of HbF, and the HbF has got increased affinity of the to the oxygen. Why? Because it is because of the 2,3-BPG. The 2,3-BPG is normally in the HbA. In normal adults, it is the HbA that is predominant. The HbA will come to the HbA. This 2,3-BPG can come and bind. If 2,3-BPG can come and bind, it can result in the release of the oxygen. Okay, but in the case of HbF has got gamma chain. So the gamma chains, into this gamma chains, the 2,3-BPG cannot bind. As it cannot bind to this gamma chain, the oxygen affinity will be high. Okay, how will we diagnose this hemoglobin? Different types of hemoglobin is that. Normally, the oxyhemoglobin, when looked through, I showed you a picture of the spectroscope. Okay, when I talked about the porphyria, when looked spectroscopically, normal oxyhemoglobin has got two bands at 540 nanometer and 580 nanometer. So when sodium hydrosulfide, a molecule, if it is added, if a compound is added to that blood, the deoxygenation will happen. So deoxygenated hemoglobin has got a color purple. Okay, and when looked through the spectroscopically, it will show only one band. It is at 565 nanometer. If you can't remember the nanometers, you can simply write in spectroscopically, oxyhemoglobin has got two bands, whereas the deoxyhemoglobin has got one band only, and the color of the deoxyhemoglobin will be purple. Okay. Transport of carbon dioxide also, you can go and read three forms: dissolved form, isohydric transport, and asparagine hemoglobin. Buffering action, I told you, it is due to the histidine, 38 histidine residues are there. It is due to the presence of histidine residues in the hemoglobin. Alpha and beta chain, the buffering action is possible. Coming to the fetal hemoglobin, it is also, I think it was discussed in the physiology classes. I'll just rush through the topic. So it will have the fetal hemoglobin, it is mainly HbF. So they have increased solubility of deoxy HbF has increased solubility, and electrophoresis, I think you will study in the coming classes. Electrophoresis is a separatory technique. Was it told in your previous classes? Separatory technique. Was it discussed? Electrophoresis?
Yes, ma'am.
It was discussed. Okay, so no need to discuss. So electrophoresis, it can separate different proteins depending upon the charge to the mass ratio. Charge to mass ratio. Okay. By depending upon the charge to mass ratio, we can separate the different components from the blood. So if RBC, if the blood is electrophoresed, we can see this HbF, HbA, HbA2, etc. and other abnormal things like HbS, HbC, etc. So normally in alkaline medium, if electrophoresis is carried out at alkaline medium, this HbF is present. HbF is normally present in very few amounts only. So this is the anode. This is the cathode. This is the point of application of the blood. So HbA has got the max mobility, means movement towards this anode. This is the anode. Okay. So HbA has the maximum anodal mobility. So you can see a large band at this end, that will be the HbA. So if HbF is present, they will have slower mobility than HbA. Okay, you understood?
Yes, ma'am.
Okay. And they will have an increased resistance to the alkali denaturation. And I told you there is decreased interaction with 2,3-BPG because gamma to the gamma chain, it cannot bind. This 2,3-BPG cannot bind. So that is why the oxygen affinity is high. And HbF is extracted by alkali denaturation. First, alkali denaturation has to be done, and then electrophoresis is done. So then we can see a slowly moving band than the HbA. Okay. Then we will move on to a different topic, hemoglobin derivatives. It is a small topic. You don't need that much detail into the derivatives of the hemoglobin. What is this derivatives is that the combination of different ligands with the heme. It is called the hemoglobin derivatives. To the heme, different ligands can be attached, or it can be due to change in simply the oxidation state of the iron. Iron is normally in what state? Ferric or ferrous?
Ferrous.
Ferrous.
So this is in ferrous state normally. If it is oxidized, the oxidation state is changed from Fe2 plus to Fe3 plus. It can also result in a hemoglobin derivative. Either it can be simply a combination of a different ligand, or it can be a change in the oxidation state of the ion. Normal blood has only two types of hemoglobin, oxyhemoglobin and deoxyhemoglobin. If oxygen is the ligand, that is oxyhemoglobin. If oxygen is not there, the hemoglobin will be combined to the water by HOH bond. Okay, this is the deoxyhemoglobin. This is the oxyhemoglobin. No, normally only these two things are there. And the deoxyhemoglobin, most of the hemoglobin should be oxygenated so that the proper supply of the oxygen has to reach all the tissues. So normally only less than 5% of the deoxyhemoglobin should be present. If the level increases more than 5%, it will lead to cyanosis. Cyanosis means like jaundice. Jaundice is a yellowish discoloration. Cyanosis is the bluish discoloration. It is not a bright blue. It is not. It is a pale, some dull color blue. Bluish discoloration seen on the nails, fingertips, lips, etc. Okay. So different kinds of hemoglobin. Hemoglobin attached to different ligands will impart different color to the blood. If it is a well oxygenated blood, the blood color will be dark red. If the concentration of deoxyhemoglobin is normally only less than 5% of deoxyhemoglobin should be there. If the concentration is more, the blood color will be purple. Methemoglobin. I'll discuss what is methemoglobin. If methemoglobin percentage is high, the blood color will be dark brown. Okay. If carbon monoxide is attached to hemoglobin, it is called carboxyhemoglobin or carbon monoxyhemoglobin. If it is there in higher concentration, it can impart a cherry red color to the blood. And if sulfur containing compounds, like if on intake of some drugs like sulfonamide, the sulfur group can attach to this hemoglobin, forming the sulfhemoglobin. If it is a sulfhemoglobin which is present in higher amount in blood, it will impart a green color to the blood. Just by looking at the color of the blood, an experienced technician can say what hemoglobin, what ligand is attached to the hemoglobin. Okay. So first we'll move on to the first derivative, which is the carboxyhemoglobin or carbon monoxyhemoglobin. Simply the carbon monoxide is attached to the hemoglobin, and you can see normal binding is with the oxygen. So 200 times affinity is more for this carbon monoxide. Okay. And it will bind with high affinity. Carbon monoxide is present, it will bind to high affinity, and it cannot liberate oxygen. Due to this high affinity, it cannot liberate oxygen to the tissues. The function of this hemoglobin is to transport the oxygen to the tissues. If it is not reaching, the oxygen is not reaching the tissues, it can result in tissue hypoxia. So this carbon monoxide, it is unsuitable for the oxygen transport. Carboxyhemoglobin will not deliver the oxygen to the tissues. Okay. So when carbon monoxide is present, see, the carbon monoxide can easily enter and reach near to the RBC, and it can bound to the hemoglobin compared to the oxygen. Okay. The causes of carbon monoxide poisoning is that it can be breathing through the automobile exhaust in a closed space, or it can be from AC, etc. Carbon monoxide toxicity, will tell it carbon monoxide toxicity when the concentration of the carboxyhemoglobin in the blood exceeds 20%. What will be the symptoms like? There will be headache, nausea, breathlessness, vomiting, chest pain, etc. Treatment is just giving the pure oxygen, administer the oxygen. It can be a fatal thing. It can lead to the death of the patient if not treated. Next ligand is the methemoglobin. Next derivative is the methemoglobin. I told you either one ligand can be attached to the hemoglobin, or it can be a change in the oxidation state of the ion. In methemoglobin, the oxidation state, the ion, normally it is the ferrous, it is changed to ferric. So methemoglobin is the change in oxidation state of the ion. And normally some ferric ions are formed in the body. Ferrous is converted to the ferric in the hemoglobin normally, but there are enzyme systems that will reconvert it back to the ferrous form. So these are the three systems which will convert. If some methemoglobin are formed in the body, it can be reconverted with these enzyme system of these 75% by NADH dependent methemoglobin reductase system. This is an enzyme which will convert the methemoglobin to the normal hemoglobin. It is with the help of a cytochrome B5. So NADH dependent methemoglobin reductase is the one which will convert the methemoglobin to the normal hemoglobin, and 20% is with the help of NADPH dependent enzyme system. So NADPH is very important for the conversion of methemoglobin to normal hemoglobin. So if NADPH is deficient, it can result in methemoglobin. Okay, and some glutathione dependent enzyme reductases there. Glutathione dependent methemoglobin reductase enzyme that can convert 5% of the methemoglobin to the normal hemoglobin. Okay, so methemoglobinemia normally less than 1%. The enzyme system, 3 enzyme system will convert back into normal hemoglobin, but there will be some 1%, less than 1% will be normally present in the blood. That is harmless. When there is increased methemoglobin, it can result in methemoglobinemia, or it is manifested by cyanosis. The bluish discoloration of the fingertips, nails, lips, etc. will be seen, which is called the cyanosis. This methemoglobinemia, either it can be congenital or it can be acquired. Congenital means it can be 3 enzyme systems are there, right, which will convert. If there is some defect in the synthesis of any of these 3 enzyme systems, it can result in congenital methemoglobinemia. Acquired means it can be due to some drugs or toxins, which will inhibit this enzyme system. Are you clear? Are you listening?
Yes, ma'am.
Yes, ma'am.
Okay. So this is the cyanosis, some bluish discoloration. Don't expect some bright blue color. So there is a dull, some dirty color. Like this will be the cyanosis, and it can be appreciated only in a fair skin person. If it is a dark skin, we cannot appreciate this bluish discoloration. So congenital causes, it can be an enzyme deficiency. This is the cytochrome B5 reductase. It was the first enzyme which will convert the 75% of it. Okay, so if there is an enzyme deficiency, it can result in congenital methemoglobinemia. Treatment is by giving a dye. The methylene blue can be effective, and the vitamin C can be effective in converting the methemoglobin to the normal hemoglobin in persons with congenital methemoglobinemia due to this enzyme deficiency. And the second thing is the acquired one, the water, the pipe water which contains high amount of nitrates or aniline dye, or some drugs like acetaminophen, paracetamol, or this nitroprusside is a drug used for treating this heart disease, especially this MI, phenacetin. All these drugs, when high amount of this reaches the blood, it can also result in toxic methemoglobinemia. Okay, and G6PD deficiency, glucose 6-phosphate dehydrogenase deficiency. That NADPH is formed from, majority is formed from the HMP shunt pathway. Okay, so when this glucose 6-phosphate is the product intermediate which is seen in the HMP shunt, from this glucose 6-phosphate only we will get the rest of the things of the HMP and we will get NADPH. So when this product is deficient, the NADPH synthesis will not be sufficient and there will be decreased NADPH. So the second enzyme system, you remember the second enzyme system which is responsible for the conversion of methemoglobin to hemoglobin was the NADPH dependent system. So when NADPH is deficient due to decreased amount of glucose 6-phosphate, it can also result in methemoglobin. You understood?
Yes, ma'am.
The same thing, the leukomethylene blue can be given as a treatment. That much you don't need. Okay. So glucose 6-phosphate is converted to 6-phosphogluconate. In that step, we will get NADPH. So when this product or this enzyme is deficient, NADPH synthesis will be deficient. NADPH is required for the conversion of the methemoglobin to the hemoglobin, normal hemoglobin. So if it is deficient, this thing will not be functioning and methemoglobin level in the blood will be high. And there is a thing called cyan methemoglobin. When this methemoglobin is combined with another ligand, cyanide, it can result in cyan methemoglobinemia. And sulfhemoglobinemia, I told you, hydrogen sulfide will be attached to the hemoglobin. It is after some therapy with drugs like sulfonamide, phenacetin, acetanilide, dapsone, etc. When these drugs are taken for a long time, the sulfhemoglobin, hydrogen sulfur, these are the sulfur containing compounds, this form. Hydrogen sulfide will come and attach to the hemoglobin to form sulfhemoglobin. Glycated hemoglobin, I think it was covered in the carbohydrate metabolism by Dr. Sajeevan. Sir has taken what is glycated hemoglobin. Right. What is glycated hemoglobin? You have to tell what is glycated hemoglobin?
HbA1c.
HbA1c. What is it?
Glucose dash to hemoglobin.
It is a non-enzymatic addition of glucose to the hemoglobin. Okay. Importance of the glycated hemoglobin, the normal values you should know. Okay. Then coming to the hemoglobin variants, I will just start this hemoglobin variants, continue with, in the next class. Hemoglobin variants. So we have seen the derivatives. Derivatives means different ligands attached, or it can be change in the oxidation state of the ion. Change in oxidation state of the ion, it is seen in methemoglobinemia. Okay. Coming to the hemoglobin variants, means some change in the chain. Polypeptide chain change is called hemoglobin variants. It can be classified into hemoglobinopathies and thalassemia. And depending upon the clinical symptoms, there is another classification. 5 types are there. So what we will see what is hemoglobinopathies and thalassemias. So I'm taking the normal hemoglobin HbA. HbA has got 2 alpha chains and 2 beta chains. I told you alpha chains are 141. It is consisting of 141 amino acid and the alpha and the other chains. They are constituting only about the all of the others. All other chains are constituting about 146 amino acids. Okay. Okay, alpha is 141. Sorry, the other things are 146 amino acids. Okay. So abnormality. So there is a sequence. There are 141 first amino acid, second amino acid, third amino acid. Like that, there are 141 amino acids. If there is a change from normally there is an amino acid at the third position. If that amino acid is changed to another one, that can result in hemoglobinopathies. Okay. Mostly this hemoglobinopathies are due to the change in the beta chain. Mainly the beta chain is affected. Any chain, any amino acid at a specific position change from one amino acid to another can result in that hemoglobinopathies. But in total, there will be 2 alpha and 2 beta. The number of alpha or beta will not change, but in one chain, the 1 or 2 chains, the one amino acid at a specific position is changed to another one. You understood?
Yes, ma'am.
Okay. What is the thalassemias is that actually the alpha chain, beta chain, the whole amino acid sequence will be changed. Same. There will be no change in the sequence like hemoglobinopathies, but the rate of synthesis will be defective. So normally 2 alpha and 2 beta will be there. Sometimes the alpha will not be produced, only the beta will be there. So chain will be chain and amino acid will be same, but some chains will not be formed. Okay. If beta chain is not formed, it can result in beta thalassemia. Depending upon which chain which is absent, we name the thalassemia. If beta is absent, we can call it as beta thalassemia. If alpha is absent, we can call as alpha thalassemia. Understood? So don't confuse. If it is a change, abnormality in the sequence, it can result in hemoglobinopathies. If it is the abnormality within the rate of synthesis of the globin chain, either alpha or the beta, it can result in thalassemia. Is it clear?
Yes.
Yes,
Okay.
ma'am.
So these are the classification based on the clinical symptoms. There are 5 types: sickle syndrome, unstable hemoglobin, hemoglobin with abnormal oxygen affinity. Okay, one more thing. Structural variations. Structural variations means thalassemia and non-symptomatic. We'll discuss each one in the coming class. So let's wind up here. I hope everything is clear for you. Any doubts?
No, ma'am.
Okay. I think most of you are engaged in some other business. Only a few are listening.
No, ma'am. No, ma'am.
Okay. When it was asked for the practicals, no one is answering anything about this heme catabolism, jaundice, bilirubin, etc. You should read today itself. You should read after the class itself, that day itself. Otherwise, you won't remember. Okay. Next class, I'll be asking questions. Read and come. Thank you.
Thank you, ma'am.
Thank you, ma'am.
Thank you, ma'am.
Thank you, ma'am.